Hello, my name is Anna Liu. I am a high school student at Appleby College, and today I will be presenting newly identified conserved domains in genes causative of intellectual disability. Let's start with some background context for the study. So in the human body, genes are responsible for creating proteins, and proteins are essentially large biomolecules or macromolecules made up of various amino acids. Proteins are also largely responsible for biological functions in our body, but when mutations happen to a certain part of a protein, they can ultimately result in diseases within a specific organ, such as the brain. So it becomes important for scientists to be able to identify the section of a protein sequence that might have experienced mutations or change, or responsible for a large, crucial biological function in our body. So in our study, we decided to focus on discovering conserved domains in protein sequences related to intellectual disability. Intellectual disability is a common brain disorder that affects 1. The intellectual abilities of an individual, which limits one's ability to learn and to think. 2. The adaptive behavior of an individual, which limits one's communication and coordination skills. ID is fairly common brain disease, and around 6.5 million people in the US are diagnosed with the disease, just around 1.9%, while just under 200 million people are diagnosed around the world with ID. It is a troubling phenomenon for those with the disease, and unfortunately, there is no effective drug available to cure this ID fully yet. Jumping into our project, the 3 main goals we had throughout the research process was A. To identify conserved domains in protein sequences that are likely causative of intellectual disability. Domains are regions of a protein that has a specific biological function and can usually function independently of the rest of the protein. B. To perform homology analysis and see whether or not these ID genes are conserved in mice and rats. And lastly, C, to identify superfamilies of these genes and look for interactions between them if possible. All these information will provide us with a better understanding of biological causes of intellectual disability, as well as more information about the specific group of genes. The general process used in this research is shown here in the diagram. The entire process is dedicated to finding the conserved domains within protein sequences of ID candidate genes. We would like to thank the creators of these software programs to help us obtain the results for our research. To break down the method, we first manually selected 2,066 candidate genes from published studies that have proven them to be causative of intellectual disability. This process is manually done, and the studies are cited at the end of this presentation. Next, we fed the list of the gene symbols into David Bioinformatics database, which separated them into clusters. In order to narrow our list and to eliminate those loosely associated genes, we limited our data within the top 10 clusters with the Go term neuron and significant p-values. As a result, we ended up with a list of 91 ID candidate genes from cluster 7, as seen in the chart below. Next, we annotated the list of genes for their biological functions and expressions in organs. Using the GTEx database for values, we narrowed our list even more to the top 10 genes that were shown to be highly conserved in brain tissues, as seen in this chart. Evidently, the later genes were significantly less expressed in brain tissues compared to the top 10 genes. And out of the top 10 genes, we calculated for their average expression value of brain tissues and found ACTB to be highly conserved in all brain tissues and significantly more than the remaining 9 genes as well. The list of top 10 genes are listed on this slide here. Lastly, we obtained the protein sequences of the top 10 ID genes from Ensemble Biomart and fed them through muscle alignment to find conserved domains. We viewed the results using GelView that clearly showcases the conservation value and qualities of the domains. Below is a screenshot of the first highly conserved domain in the sequence alignment, which goes to show its high potential for being correlated to intellectual disability. With our list of genes, we also performed a homology analysis, and all 10 genes were conserved in both mouse and rats. But what's more interesting is that when we classify these genes by superfamilies, we realize that GNAO1 and RAC1 belong to the same superfamily, CL3A936, as seen by the BLAST-P results in this chart. Using string, we also explore the protein interactions of these genes. And the same two genes, GNAO1 and RAC1, interacted with each other with a combined score of 0.924. The diagram here is obtained from string, and it shows the first layer of GNAO1 protein interactions, which is a very unique and interesting result. And in conclusion, we believe that our workflow can be valuable for identifying conserved domains related to specific diseases. In the future, we look to identify motif and variants within the protein sequences, which might be causative of intellectual disability. Also, the ability to identify conserved domains and motifs can be helpful in guiding a drug design process called the structure-based drug design, which looks for functional sites in proteins and to fit drug compounds into the active site pocket. If successful, this can lead to the development of long-needed drug for specific diseases. I would like to thank the individuals listed here for their contributions and guidance during the research, as well as my parents for their constant support.